We're going to watch a little video that walks us through all the phases so we can see what happens and how meiosis is different than mitosis.
Imagine a diploid cell during interphase. The chromosomes are long, thin strands of DNA within the nuclear envelope. There is a single pair of centrioles near the nucleus. Near the end of interphase, the cell prepares for the first phase of meiosis, meiosis 1. The centriole pair duplicates. Each chromosome is replicated. Homologous chromosomes begin to group together. Meiosis 1 begins. The first phase of meiosis 1 is prophase 1. During prophase 1, the centriole pairs move to opposite sides of the cell. The chromosomes condense. The homologous chromosomes are tangled together. A total of 4 chromatids are present, since each chromosome was replicated before meiosis began. Each pair of sister chromatids is joined at a centromere. While the pairs of chromosomes are close together, they may break and exchange segments. This is called crossing over. The nuclear envelope dissolves and the spindle fibers form. The homologous chromosomes remain paired as spindle fibers pull them toward the center of the cell. The second phase of meiosis 1 is metaphase 1. During metaphase 1, the homologous chromosomes line up in the center of the spindle. The third phase of meiosis 1 is anaphase 1. During anaphase 1, the homologous chromosomes are pulled apart by the spindle fibers. They move to opposite ends of the cell. The fourth phase of meiosis 1 is telophase 1. In some cells, the nuclear envelope forms around the chromosome sets. Cytokinesis begins when the cell membrane starts to pinch the cell's cytoplasm in half. 2 new daughter cells are formed. Each daughter cell is haploid. Each has half the chromosomes from the parent cell. In preparation for another cell division, the centriole pairs duplicate. The chromosomes do not duplicate. Each daughter cell is now ready for meiosis 2, the second cell division of meiosis. Meiosis 2 is very similar to mitosis. However, meiosis 2 starts with a haploid cell. The first phase of meiosis 2 is prophase 2. During prophase 2, the centriole pairs move to opposite ends of the cell. The nuclear envelope dissolves and a spindle forms. The chromosomes are pulled toward the middle of the cell. During metaphase 2, the second phase of meiosis 2, the chromosomes line up in the center of the spindle. In anaphase 2, the centromeres of the sister chromatids separate. The chromatids are moved to opposite ends of the cell. Each sister chromatid is now considered an individual daughter chromosome. The last phase of meiosis 2 is telophase 2. During telophase 2, nuclear envelopes form around the chromosomes. In cytokinesis, the cell membrane pinches the cell in half. Each haploid cell has now divided into 2 haploid daughter cells. The original diploid cell has given rise to 4 daughter cells, each with a single set of unduplicated chromosomes.
We watch this video now, and let's take a look and kind of walk through our phases to kind of see what the sequence is and what's happening. So we're going to start with our cell in interphase 1, and cell is hanging out in a testes or an ovary. Remember, these are going to happen in reproductive cells now, this kind of cell division. We're going to copy our chromosomes, go through S phase, and now the cell is ready to do meiosis. We go into prophase 1. Each of our homologous partners are going to buddy up to make a tetrad. Metaphase 1, go to the middle of the cell. Anaphase 1 is apart. Our chromatids are going to stay together, but our homologous partners are going to go apart this time. In telophase 1, I see 2 nuclei. Cytokinesis is going to split the 2 daughter cells apart, and then each one of those is going to go on to divide again. Prophase 2, metaphase 2, go to the middle. Anaphase, A for ana, A for apart. Our chromatids are going to go apart. Telophase 2, I end up with 4 haploid daughter cells. So let's kind of compare side by side and look at what's happening in what stages, what phases are different. We're going to start with an interphase cell. We're going to put mitosis on the left here and meiosis on the right, so we can kind of compare as we walk through to see where those differences are. In interphase of mitosis, our DNA is spread out as chromatin. We're hanging out in the body. We're doing our job. We have chromatin open so I can read it and know what I'm doing. Our nuclear membrane is visible. If I look at an interphase cell under the microscope, I see a nucleus, I see a nucleolus. We're going to copy our DNA and S. Remember, S and G2 are part of that interphase. If the cell is going to divide, it copies its DNA and S and makes all the molecules and cell parts we need for the new cell in G2. And that same thing is going to happen for a cell getting ready for meiosis. In interphase 1, all of those same things are true. We don't have to learn a new phase. We're going into prophase. We scrunch up our chromatin into chromosomes to be easy to move. We lose our nuclear membrane and our nucleolus so that we can move the chromosomes around. They have room to move. And our spindle, our centrioles, chromosome moving machinery appear to pull the chromosomes to opposite ends. And those same things are going to happen in prophase. We don't really have to learn a new phase, except for one thing. Here's the first difference. Homologous pairs are going to match up in prophase 1. Partners go together and form those tetrads, and it's going to allow those chromosomes to start crossing over, start becoming different, trading DNA between homologous partners. M for meta, M for middle, chromosomes line up in the middle. We're going back to the middle again, only now look at the difference here. Chromosomes line up independently. Those homologous partners don't really care what each other are doing in mitosis, but they're going to buddy up, and because we did synapsis, match up the partners in prophase 1, now those partners, those homologous buddies, are going to go together with their buddy to the middle of the cell. So we're still doing M for meta, M for middle. A for ana, A for apart. When we did anaphase of mitosis, chromatid arms split and each one went to a different end of the cell. Now what's going to go apart is our homologous partners. Notice this chromosome still has 2 chromatid arms. So apart again, yes, A for ana, A for apart, but chromatids are going to stay together. What's going to go apart is the homologous pairs. The homologous partners are going to split. T for telo, T for 2. I can see 2 nuclei. Nuclear membrane returns, nucleolus returns, the DNA goes back. Remember, this is called reverse prophase. We're resetting the cell. We lose our spindle and our chromosome, excuse me, pulling machinery. Telophase number 1, same as mitosis. All of those things are going to happen again. So we don't really have to learn a new phase. Some cells actually put the nuclear membrane back. There are some cells that kind of do a little semi-reset and don't totally return the cell back to the way because we're going into another phase. But basically, nothing different is going to happen in telophase 1. Cytokinesis, we're going to split the cytoplasm. Cytoplasm splits into 2. We're going to do that pinch, cleavage furrow. If I'm an animal cell, make a cell plate. If I'm a plant cell. And cytokinesis number 1, same as mitosis. Nothing new. We don't have to learn a new phase here. We're going back into interphase now. If I was a mitosis cell going back into interphase, the DNA spreads back out as chromatin. My nucleus returns. If this cell was going to go and divide again as mitosis, we would copy our DNA the second time before we went back into the cell cycle. And here's our last difference. We're going to skip interphase 2. The DNA is not going to get copied this time. So we're going to skip this phase. We go right back into cell division, and we're going back into prophase number 1. Or excuse me, prophase number 2. So same as before, DNA scrunches into chromosomes, we lose our nucleus, centrioles and spindles show up to move the chromatin, our chromosomes around, and we are going to do that exact same thing in prophase number 2. We have chromosomes scrunched up, no nucleus so that we can move our chromosomes around, and spindle, our centrioles, centrosome area is going to organize to hook our chromosomes onto spindle fibers just like it did in prophase number 1, at prophase of mitosis. M for meta, M for middle, chromosomes line up in the middle, same as before, we're going to the middle. We have fewer chromosomes now because we cut our number in half from that first division, but everybody's going to line up in the middle of the cell. A for Anna, A for apart, chromatid arms are going to separate just like they did in mitosis, anaphase. Nothing new. We don't have to learn a new phase here. You're kind of starting to see the picture. T for tello, T for 2, guess what? Nothing new. Those same things that happen in telophase that you know from mitosis are going to happen in telophase 2. So, cytokinesis, we do the pinch thing, cleavage furrow. If I'm an animal cell, cell plate, so make that little dividing wall. If I'm a plant, cytokinesis 2, same as mitosis. So think about those differences. All of the differences that we saw comparing mitosis and meiosis really happen in the first time through, the first meiosis division. The second time we divide, prophase 2, metaphase 2, anaphase 2, telophase 2, cytokinesis 2, they're basically a mitosis division. We think about it. All those differences that happen really happen during the first meiosis. Prophase 1, matching up our homologous partners and trading DNA, that synapsis and crossing over. Independent assortment and segregation at anaphase 1, mixing up the mom and dad chromosomes in different combinations, separating those homologous partners are going to allow more genetic recombination. And then skipping interphase before I go into meiosis, the second division. Divide twice, but I'm only copying the DNA one time. Most of those differences are at that first division. The second meiosis, prophase 2, metaphase 2, anaphase 2, telophase 2, cytokinesis 2, they're basically a mitosis division. So there's only really a few phases that we need to learn that are different. We need to talk a little bit about how making sperm and eggs, though, are different. And so, vocab word, spermatogenesis, making mature sperm. They mature. Once I do my meiosis to produce those 4 daughter cells, they mature by growing their tails. Remember we talked about flagella in our cell parts chapter. They have to be able to swim to the egg. And basically, if you look at it now, testes cell is going to divide to make those 4 daughter cells. I get 4 little spermatids from every meiosis division. If you compare the size of a sperm compared to an egg, the sperm is a lot smaller. That sperm head mainly just has chromosomes. It's a nucleus with chromosomes inside, very little cytoplasm. And the reason is we want that sperm to be small. We want it to be lightweight so it can swim fast and go far. It's got to be able to reach the egg. And so very little cytoplasm in the head of a sperm, mostly just DNA. The dad is donating the chromosomes. Most of the cell parts now, the starting material, the nutrients, the organelles, all those molecule building blocks, we talked about proteins and carbs and all of those molecules from our first chapter that are going to provide the start for the baby is really coming from that egg cell. And so because we want to have the best start for the baby, the most cytoplasm available for that starting cell, egg cells, the ones when we're doing meiosis to produce egg cells, is a little bit different than the meiosis that makes sperms. When we're making eggs, oogenesis, it's a kind of funny word. It's got double O's in front. An oocyte is an egg cell. And it's not an oo, and it's not an oo. It's kind of a run together, oogenesis. We're making eggs. And notice the difference now. Because we want that egg cell that's going to provide the start for the baby to have the most nutrients, the most molecules, the most cell parts possible, when that ovary cell divides the first time in meiosis number 1, all the cytoplasm goes one way. We make one really little tiny cell that mainly just has chromosomes. The chromosomes are going to divide evenly, but the cytoplasm does not. Mostly this little cell is just chromosomes with a very little bit of cytoplasm, and all the cytoplasm goes one direction to one of the daughter cells. And the same thing is going to happen. Now we do meiosis number 2. That little cell is going to divide again, but really doesn't have very much cytoplasm to share. It's going to make 2 small cells, and all the cytoplasm is going one way to make one big giant egg. And these little guys, 3 little, we call them polar bodies. The cytoplasm, when we're making eggs, is going to divide unevenly. So we end up with one really, really, really giant egg cell. It has all the nutrients, all the cell parts, all the best start for our baby, and 3 little polar bodies that are mainly just chromosomes. So think about sperm mostly is donating the DNA. All of the cell parts, all the nutrients are coming from that egg. We're making one really, really good egg to give the baby the best start. And that's the reason why, even guys in this room, all of your mitochondrial DNA come from the mother. They come from the egg. And it's because the egg is donating all those cell parts and the nutrients to get the baby started. So what do the polar bodies do? Remember, we talked about self-digesting for the good of the organism. We talked about cell parts in our cell parts chapter. What's our cell part? That is the sac of digestive enzymes that break down unwanted cells. We're going to use lysosomes again, and those digestive enzymes are going to self-destruct the cell. Those little polar bodies do that apoptosis thing. Cell suicide for the good of the organism. They are self-destructing so that we can give all the cytoplasm to one really good egg for the best start possible for our baby. That's the end of meiosis, and thanks for watching.